Good day. In this colloquium, I will be presenting my study titled Designing Potential Cancer Therapeutics for Clusterin-Induced Chemoresistance Using Fragment-Based Drug Discovery. I am Angelo John Gabriel Vicar, a student under the joint program Bachelor's and Master's in Biological Engineering. This project is under the supervision of Professor Lemuel Tayo. Cancer is now one of the leading causes of death worldwide, accounting for nearly 1 in 6 according to the World Health Organization. And despite the amount of data and the amount of studies we have conducted on cancer, we have yet to possess the necessary solutions required for us to deal with this issue. One of the complications in this situation is that of chemoresistance. It is a phenomenon in which cancer cells develop mutations that allow them to shrug off treatments such as chemotherapy. One of the main proteins responsible for chemoresistance is a molecular chaperone called clusterin, also called apolipoprotein J. In normal cells, it functions as a lipid transport, membrane recycling, and cell adhesion protein. However, crucial for our study for today is that of its role in programmed cell death or apoptosis. Crucially, there are two forms of clustering which were determined to have antiparallel activities. The first is the secreted clusterin and the nuclear clusterin, which are responsible for anti-apoptotic and apoptotic functions respectively. As of the time of this study, current treatment options for clusterin primarily involve antisense oligonucleotides, such as that of OGX-011, which are often used in conjunction with chemotherapy drugs such as gemcitabine. Now, there are indeed disadvantages with this treatment method. For one, they are easily degradable without proper drug delivery systems, and thus it increases their production costs. And there are also issues relating to dosages, because we are talking about knocking down specific sections of the genetic code. Now, this indicates the need for a potential alternative form for clustering inhibition. One of the modern drug discovery pipelines which are the most prevalent is that of fragment-based drug discovery, which makes use of small molecules called fragments which obey the rule of three as stated in this slide. Now their small size allows them to fit in the binding pocket very snugly, and from then on, they can be linked, they can be grown, and they can be merged into proper, novel, and fit candidate drugs whose ADMET properties or toxicity properties can be properly optimized. In addition, they can also be used to probe the protein for possible binding spots, which can be used for alternative or allosteric inhibition. And thus, the aim of this study is to construct and design novel candidate drugs for the potential inhibition of secreted clusterin using fragment-based methods. It also seeks to describe the potential binding sites of clusterin. And finally, it aims to determine the structural qualities that highly correlate with that of the binding affinity to secreted clusterin. And now for the methodology. First, we made use of commercially available fragment databases, which then went through filtering using the rule of three and anti-cancer activity prediction using PDCSM web server. The final fragment database, composed of 607 potentially anti-cancer fragments, were then docked to the crystallized structure of secreted clusterin or SCLU obtained from RCSB PDB. Hotspot analysis from FTMap was also utilized to validate the binding site description. From the binding affinity ranking, we obtained the top 1 fragment, which then underwent fragment growing under Frago web server using the hydrogen, substructure, and retrosynthesis based replacement algorithms. The potential drugs were then assessed for their predicted toxicity using test software, only selecting for non-mutagenic and developmental non-toxicants. This gives us a candidate drug database composed of 194 candidate drugs. The compounds were then docked again. This data would form the basis for the QSAR model development. ADMET properties were then used to pick the top 1 final candidate drug, which then underwent 3 trials of 100 nanosecond molecular dynamic simulation with the co-crystallized ligand NAG as the reference. We can see that the chemical space covered by the fragment database was small as evidenced by the normalized RMSD values of the main and final fragment databases, which are 0.0364 and 0.032 respectively. This is a given fact with fragment-based methods, as the lower chemical space are used as the building blocks to expand the coverage of the dataset. From the blind docking and FTMap results, we can see that they both agree on a potential main ligand binding site, featuring amino acids such as serine 161 and arginine 158. Interestingly, the other end, featuring residues such as isoleucine 46 and leucine 211, indicate potential allosteric or protein-protein interaction sites. Looking at the binding affinity, we can see that the range of the binding scores is relatively low from negative 3.2 to negative 8 kilocalorie per mole. This is because the smaller fragments do not have the size to properly interact with the amino acid residues, an issue that is solved using the fragment growing methods. With only one round of fragment growing, we can see that there is a great improvement in the binding affinity, now reaching up to negative 10 kilocalories per mole, and a mean and median affinity of approximately negative 8 kilocalorie per mole. With the principles of fragment growing, we can also address the potential toxicity risk highlighted by test by using subsequent optimizations in the chemical structures. In order to determine the relationship between structure and the binding affinity, we make use of quantitative structure activity relationship analysis, or QSAR. In this case, artificial neural networks of up to 3 hidden layer architectures was used. The model with the highest test score has a hidden layer size distribution of 20, 20, and 130 neurons for each respective hidden layer. This produced a model with acceptable predictive ability as evidenced by the test score of 0.63, a relatively close cross-validation mean of 0.56, and a normalized RMSE of 12.78%, which is close to the accepted threshold of 10% according to the literature. If we look at which chemical descriptors contribute significantly to the binding affinity, we can see that molecular complexity, represented by average IPC, and the degree of branching, also known as Balaban J, were most relevant. This indicates that for molecular chaperones, such as clusterin, the individual variations do not matter as much as the overall complexity of the molecule to cover as much of the binding pocket as possible. If we look at the applicability domain for which the model is fit for, we can see that four distinct clusters form due to the type of alterations conducted from the fragment growing step. It can be seen that cluster 2 features less complex substructure replacements as indicated by the lower physical measurements and the lower binding affinity. Cluster 0, meanwhile, we found are composed of candidates whose alterations are present in the peripheral pyridine and pyridazine substructures. Lastly, the closely related clusters 1 and 3 were found to be the results of modifications in the central pyrrole analog. In our selection for the top final candidate drug, we considered only the general case, meaning the drugs must be non-blood brain barrier permeant. They must be at least moderately soluble in lipids and in water, and not inhibitors or substrates of CYP1A2 and CYP3A4. This ensures minimal cytotoxic clashes with other drugs. The final distinguishing factor is the environment of SCLU, which requires a low consensus log P, which ensures proper activity outside the cell without interfering internally. Under the molecular dynamics simulation, the top one final candidate drug produced remarkably close results with the reference ligand, although the RMSD and radius of gyration were slightly higher for the candidate drug with means of 5.19 and 34.28 angstroms respectively, as compared to 4.8 and 34.12 mean angstroms of the reference ligand. This indicates the complex of the drug and the reference are close in terms of structural stability. The RMSF data for the top 1 final candidate drug shows a possible explanation to the higher values. Due to the size of the candidate drug, the structure moves around a bit more, as evidenced by the flexible peripheral substructures. These assertions can be supported by the backbone RMSF data, which shows remarkably similar results for both complexes. Energetically, however, the drug complex showed higher binding affinity from MMGBSA calculations with a mean of negative 34.01 kilocalories per mole as compared with the reference having a mean of negative 12.16 kilocalorie per mole. The Cliffs delta effect size was found to be negative 1, which shows a significant difference between the binding affinity data for both ligands. ADF test results were then used to also determine the convergence of the energy terms, from which we can see that only one run did not reach stability in the case of the drug, whereas multiple runs of the ligand remain unstable. We also docked FDA-approved anti-cancer drugs, which we found are less potent than the candidate drugs by 19% in terms of binding affinity. If we consider the druggability of molecular chaperones, such as secreted clusterin, we note that the flexible binding sites of these proteins allow them to accommodate a large variety of drugs. However, this runs to an issue of specificity, which can be addressed by properly selecting ADME properties. Synthesizability was also a strength of fragment-based drug discovery, as analysis by the DeepCA web server shows that 148 compounds scored as easy to synthesize. We also note that the pyridine and pyridazine substructures were found to be heavily involved with anti-cancer activity, according to the literature. However, the central fused pyrrolidine rings were also found to be somewhat related. However, more literature is required to support these claims. For our conclusions, we have successfully produced a database of potential clustering inhibitors who are shown to be effective under molecular docking and dynamics simulations. Additionally, we also identified the potential ligand binding site and even potential allosteric binding sites. Finally, through QSAR analysis, we determined that molecular chaperones such as clusterin favor overall complexity as compared to more minute structural differences across ligands, with the model producing acceptable predictions according to the test score of 0.63. And for our recommendations, we recommend wet lab validations for our findings, along with additional simulations to gain further insight on conformational states of the ligands, as well as further optimization by subsequent fragment growing optimization steps in order to improve ADMET properties and binding affinity. The results of this study can be the start of potential drug delivery systems, protein targeting systems, and even protein-protein interaction facilitators by only slightly tweaking the structure of the candidate drugs. Additionally, the database is also useful in other diseases wherein clustering is highly involved, such as that of Alzheimer's and other neurological diseases, with only tweaks in the ADMET properties selection. That will be it for my presentation. Thank you for listening and have a great day.